tissue engineering and in bioprinting, we use perfectly fitting and patient-specific, so highly individualized three-dimensional structures, which should fulfill a specific function in the body after transplantation. And in comparison to simple implant materials, these tissue-engineered structures, they possess a biological component, the patient-specific cells, which should ensure, for example, a better tissue integration after transplantation into the host organism, or take a specific task, for example, muscle cells in cardiovascular biohybrid implants, which should then be involved in vessel contraction.
These cells are processed in bioprinting using 3D printing. Two different methods are used at the Fraunhofer ILT in Aachen, Germany: the lift process and the stereolithographic process.
We developed mainly two different processes. One is the laser-induced forward transfer process, which is a printing process to directly print single cells or clusters of cells. And the other process is stereolithography. Stereolithography is a process where we cross-link polymers to print scaffolds, which can be used for implants. The special thing in this process is that we use new, not common laser sources, allowing us to use other polymers and materials.
One of the fields of application is cardiovascular tissue engineering. In a joint project, the Fraunhofer ILT and the University Hospital RWTH Aachen are working on the manufacture of heart valves.
The next step, if this works, is that we really print a heart valve scaffold looking like a heart valve and provide this to the clinicians, and then they try to mature the heart valve in the bioreactor, for example.
In our sub-project, we produce fibrin-based hydrogels together with our partner Professor Pisch from the DWI in Aachen. And we can modulate these fibrin-based hydrogels in a way that we can direct the cell behavior. And the partners from the Fraunhofer ILT, they produce 3D printed structures where we can add the cell-laden hydrogels, and they also developed a bioreactor, or better, a mechanoreactor, together with their cooperation partner, Professor Mehler in Munich. And with this bioreactor, we can stretch our cell-laden hydrogels. With this, we can, let's say, train the cells in the hydrogels in our cell culture lab for their future function in the organism.
In which direction could bioprinting develop in the future? The scientists already have several ideas.
The great vision today is to print one time a human being. The steps towards the human being are to print little organs, for example, which can be used on organ-on-a-chip systems. And these systems can be used, for example, for the development of medicine, of pharmaceuticals, and they can provide an insight into the connection between different organs and to understand the processes.
I feel that it would be a good alternative if we would directly use smart materials, which, for example, release in the body directly one factor which recruits endogenous stem cells from the microenvironment, and another factor which then induces the recruited stem cells to differentiate towards a specific cell fate, so that the complete constructs and the functional construct is directly produced in the body and not before in the cell culture lab.